transmitir para o YouTube agora.
Okay.
Okay. So now we are changed to English, okay? And I'm going to do a brief introduction of Dr. Otávio Cabral Marques. And first of all, thank you everyone who joined us in this immuno webinar. Today we are glad to have Dr. Otávio Cabral Marques talking about integrative systems immunology during the COVID-19 pandemic, the challenges and opportunities. Dr. Otávio Cabral Marques is biologist. He obtained his master and PhD degrees at the Institute of Biomedical Science, University of São Paulo. During his PhD, he did a sandwich PhD at the University of Washington, Seattle Children's Research Institute in USA. Otávio performed his postdoctorate in Brazil and Germany at the University of Lübeck and the University of Freiburg. Otávio also is the Brazilian Junior Ambassador of the Universal Scientific Education and Research Network. And currently, Dr. Otávio is a scientist at our department, the Department of Immunology, and also at the Department of Clinical and Toxicological Analysis, both of University of São Paulo. His research group is working with systems immunology applied to understand the molecular landscape of different viral infections. So thank you so much, Dr. Otávio Cabral Marques, for accepting our invitation. After your talk, then we are open for questions.
So thank you very much, Paula, for the invitation and all the organizers. It's really a great pleasure to be here to participate on this meeting, these fantastic seminars of immunology. I remember now when I was a student, when I look forward backwards, and I see how many years are soon gone. So I get very happy to be here now as I speak and to present the work developed by my group. This is not my work, but it's the work of several students. Paula is a postdoc and several PhDs and research collaborators, and I try to summarize here what we are developing together as a team. So as you can see, our lab is called the Laboratory of Integrative Systems Immunology. So, and I'm currently in the Department of Immunology, but also in pharmacology in USP, and now also getting associated to the FMUSP here in São Paulo. So we are studying and investigating emerging viral diseases. So as you can see here, let me just adjust here the camera here. So we are investigating currently emerging viral diseases that can be developed currently by several facilitating spreading processes, like the increase of commercial air travels and global trade, and in summary, the globalization process. They are promoting the rapid spread of several infectious diseases, and we saw nowadays 2 years ago the development of the COVID-19 pandemic due to these traits, but also certainly science and technology have developed a lot and fastly in the last years, and also we have several advances facilitating the control of these pandemics and these emerging viral diseases. As you can see, as we all saw, the rapid development of the vaccination through different new platforms were essential to control the COVID-19 pandemic worldwide. But not only the COVID-19, we have also several other diseases. Like we lived here with dengue, with yellow fever, also chikungunya, and also other viral infections like influenza that emerge and re-emerge periodically, and making necessary to understand well how these diseases, the physiopathology of these diseases, to develop new therapy and also vaccinations. And here, as you can see, there are several areas that are predicted to be of great interest, because they can probably be a focus of new and novel zoonotic viruses, as we saw with COVID-19, according to the relationship between the animals and also how the humans have grown up their cities throughout these areas, and also several habits, as you can see in these slides, that make the development of these diseases more fast and also, unfortunately, severe due to new viruses that can emerge rapidly throughout the world. So more recently we saw, as we told here about the COVID-19 that was spread by because the SARS-CoV-2 virus went from probably developing in the pangolin species and passed through the process of spillover from these animals to men. So, and then nowadays we have approximately 500 million cases that, let me only close the door here, please. So we have approximately 500 million cases, with the pandemics more controlled nowadays, but we had 6.1 million deaths worldwide. And also, as you can see, the vaccination could control this process nowadays. And then in Brazil, we had 30 million cumulative cases and 0.6 million deaths, which is pretty a lot, unfortunately, in our country. And more than 423 million vaccinations administered throughout the country. And we have nowadays approximately 162 million people fully vaccinated. And just to talk a little bit about the pathology of COVID-19, and you all probably are aware of this process, and it will be a little bit fast, because after 2 years seeing many of this process, we are aware that the spike protein is responsible for the entrance of this virus into the human cells, binding to its well-known receptor, the ACE2. And also other co-receptors are well-known, like the TMPRSS2, and several pathophysiological processes are now well characterized. Like we have, and we know about the endothelial damage that occur in COVID-19 due to this hyperinflammation process, a lot of fibrosis and coagulation process, and several dysregulation of the immune response that we will approach here. For example, we have lymphopenia of lymphopenia, like affecting CD8, CD4, NK cells, B cells. We have also T cell hyperactivation characterized by the expression of several activation markers, and also the cytokine storms that we all rapidly know in the beginning of the pandemic. And also there is the existence, the coexistence of cell exhaustion that have been characterized in COVID-19 and several other cellular abnormalities such as neutrophilia, for example, that's well known in COVID-19. And we have also here the cytokine storms that I told to you of several pro-inflammatory cytokines and simultaneously some anti-inflammatory cytokines such as IL-1 beta, IL-6, and IL-10 respectively, like IL-10, a central anti-inflammatory, and the others pro-inflammatory cytokines. And we have here also that that have been characterized autoantibody productions in COVID-19 more recently that we'll talk a little bit on during this talk. But I would like to tell you how I was starting to like systems immunology. So I remember during my undergraduate studies, I saw some professors talking about changes in the paradigms, like from the Newtonian Cartesian way of linear approaches that have been developed to new systemic, like telling, I was fascinated by Fritjof Capra. I got the books from this physics that he characterized very, very well, new way of see the life, how we can see our lives, how we can approach science, how we can, for example, there was a nice documentary that's called The Turning Point, O Ponto de Mutação, and also reading connections in very, very nice books that tell about the systems view of life. And I remember that in one of these books, Capra was telling that when a butterfly shake its wings in one side of the world, we can see a thunderstorm in the other side. And I was, oh my God, is this really possible? But I was fascinated, and I believed in that idea that nowadays we saw with the COVID-19, that's really true, that some thunderstorms from one side of the world can affect the whole world, right? And in several means. For example, now, and then we will show to you our approach, how we work, what systems immunology, how systems immunology is very important to understand how the human immune systems work. For example, we go from healthy to disease, and we have several aspects of these layers that we call transcriptome, proteome, also signalome, metabolome, and also several other aspects that we have several information, thousands of information through high throughput data, and we get this data and we started to investigate how they relate to each other, to understand not only linear signaling pathways, but to see how the interaction of these molecules are essential to understand sensibility, susceptibility, or resistance to diseases, resistance to immunotherapy, and also to develop new biomarkers. And here, for instance, we see that since from 2007, we have these investigations, these studies, they have been essential to develop what we call network medicines. For example, this seminal work was essential to show that these genes called FTO gene increases the obesity to 30%, the risk of obesity. But a person who have friends that are obese, they have even more chance to get obesity higher, like in 57%. So beyond our genes, we have social interactions, we have epigenetics, we have several other factors that we have a lot of interest to understand, to get more insights into the immune system. So here, the approach of the systems immunology that we have used. For example, you can see here, we have patients with their clinical information. We have traditionally the obtain of some ELISAs or flow cytometry data. And what we do, particularly in our lab, we go to the computers and we do bioinformatics analysis. We use particularly data integration or machine learning or several other in silico analysis and modeling, and also network analysis, and also to get translational information and also biomarkers to obtain new hypotheses, new models, to go back and to do new experiments for the development of therapy and also vaccinology, systems vaccinology, which is really important. And so what we, just one piece to show to you an example, what we do, we get, of course, original data that we can generate it, but here some examples of data that we obtain databases through these hundreds of high throughput informations that are deposited in geodataset, for example, in the geodatabase, and we get these geodatasets to try to find the hidden information through the process of data creation that we obtain this data. We go to get the transcriptomes or proteomic data. We do differential expression analysis to see genes and protein differently expressed. And then we try to see the consistency between this information throughout, across the different studies. And then we go to get functional insights, doing gene ontology analysis to see the biological process, molecular functions, cellular components, and pathways through our analysis, and to get the immunological signatures, to see stratification, for example, to see which genes are important for patients' disease severity, and also UTI, for example, the chance of patients to go to UTI. And then we see the interactome, and then we get this data to go back and to do some experiments. And here you can see this was the paper when I was in Germany doing my postdoc, and I start to get very excited to do systems immunology. I will talk a little bit about this, but more showing what the students of our lab, what they have developed. And here, some example, what we have done, some relationships between cytokines and going back and forward. And I will show you some of these results that we have obtaining during the pandemics, doing computational analysis and getting data throughout the world to develop some works here. For example, the paper now recently submitted by Gabriela, who is doing her PhD here in our lab. And here we'll show to you, for example, we started, Paula, the moderator here of this immunological webinar, she developed in the beginning of the pandemic, the COVID-19 pandemic, some investigations to explain why are males more susceptible than females to severe COVID-19. So Paula made a wonderful job, a very beautiful work together with the video here is not working somehow, I don't know why, but to see how females may be more protected than men, because what we are seeing, particularly during the beginning of the COVID-19 pandemic, was that females, they have the same ratio of infections compared with the men, but men were getting much more severe infections than women in relation to the ratio. So this was a wonderful job that Paula did together with Alexander and also Gabriela. And just to show you quickly, what Paula found was that females, they present a cytokine storm pattern that were more similar with young subjects. As you can see through these bubble heat maps, you can see that young and female patients, they have these patterns of molecules associated with type 1 interferon that we all know that's essential to control viral infections. And also simultaneously, Paulo was seeing that the signatures related to damage that were the neutrophils mediated immunity genes, they were also following this pattern. Females were clustering together with young, while male and elderly individuals were presented less downregulation of these important pathways involving damage. So I already presented data several times also in the Department of Immunology. I will go forward to the next steps, what we have found in the last month. But what Paula found was that something that's evolutionarily very important. Females, during their lives, they have to be able, for example, during pregnancy, to control well viral infections and several other infections, but they have to modulate. They have important plasticity, intrinsic plasticity capacity to avoid damage, exactly to avoid loss of babies during pregnancy. So that was really beautiful work, and this brought us up to investigate it a little bit more to understand these genes that Paulo was seeing insights, important insights to show that these genes are associated with the severe COVID-19 infection, the down modulation in blue, as we saw, while upregulation of important interferon-associated genes as a mechanism to avoid severe COVID-19 infection. So then we went to the second part of this history to characterize that severe COVID-19 has a neutrophil related signature, immunological signatures in relation to cytokines. We search these neutrophils and cytokines and chemokines association with several other inflammatory diseases like are being characterized now at MISC, HLH, and several other features of severe and damaged inflammatory diseases. Then we went to see what COVID-19 has in common with these diseases, with these features. And then we saw cytokines, several molecules associated with cytokine. We saw also several molecules associated with chemotaxis. And here, the most prevalent that we saw, that was neutrophils-mediated immunity. I will show you in the next slides in details what we are seeing here. But what we searched was a multi-tissue analysis, analyze swabs of patients with COVID-19, and also data sets from bulk or single-cell RNA sequence to understand, well, what does it mean to have several molecules in common? And then we go here, and then we saw that there are several granular associated molecules that are commonly dysregulated in COVID-19 patients. As you can see in this molecular function analysis, you see that there are several granular associated molecules, as you can see here, ranging from azurophilic, also primary granular, secondary or specific granular, tertiary granular, that are important to protect patients against infections, but they are also involved with COVID-19 damage, as it's now well known. So what we did to see which are to rank these molecules, we did, for example, principal component analysis associated with machine learning using an approach that's called random forest to rank these genes, the importance of these genes, also to select genes that are stratifying patients with COVID-19 from controls, and also patient 19 not in intensive care unit from those in intensive care unit. And here, as you can see, patients with COVID-19 using this principal component analysis with spectral decomposition, we can see, like in a vectorial analysis, that are several neutrophils associated genes pulling these patients or pushing these patients to stratify them from the other less severe disease or from controls. And then you can see here, we have arginine 1 and several other molecules that are involved, probably demonstrating an attempt to suppress the immune response, as we know about the arginine 1 function, together with the other granules associated molecules. So we found here molecules, first important to be biomarkers of these diseases and of this severe disease. And then we see here by a multi-layer transcriptomic analysis using single cell or even microarray or book RNA sequence, that these molecules are dysregulated in several data sets through several studies. We can see here from single cell and also with book that patients with severe diseases, they increase the expression. Compare, for example, the same pattern to HLH, where patients also present upregulation of several neutrophils associated molecules. And then we use single cell, we obtain a data set in collaboration with Germany to better understand which populations were expressed in genes. And then we see the expansion in this German cohort of immature neutrophils and classical monocytes also. And as you can see here, there are, wait a moment, immature neutrophils. In addition to several other populations, when you go here from mild disease controls, mild disease, and also severe in dark blue. And here, in the row 2, we see each cell population, as you can see from left to the right. And here, underneath the heat map, you can see the legends for the cell populations. And then you go to immature neutrophils, we see that the several molecules expressed during severe COVID-19, and then that the literature have associated with lung damage, thrombosis, and several other pathological processes during COVID-19, we see that the immature neutrophils are expressing highly these molecules. And these immature neutrophils, they have been classically associated with severe inflammatory diseases and also rheumatological disease, for example. And just to summarize during our time here, this first part of our work, we went in these studies that Paula and Lena were developing from more than 1,100 individuals and from more than 21,000 dysregulated genes, went in a top-down process, found, for example, 239 common DACs between several COVID-19 data sets published across the whole world, across our planet. And then we saw 25 genes stratifying COVID-19 patients in ICU with the severe diseases, and found particularly only 11 genes that are more strongly associated with this process, to summarize this multi-layer way to find new biomarkers in COVID-19 disease patients. So this data, this integrative systems analysis provides explanations, or at least insights, strong insights for the COVID-19 severity that have been mechanistically, which is not our, I'm now here to show to you, but have mechanistically proved by several other studies that are important to characterize different symptoms common between patients with COVID-19 and other severe disease. And here we go to an excited party that I like a lot also, that recently this wonderful paper by the IKICO group were showing that patients with COVID-19, they develop several autoantibodies, which are involved with the regulation, probably, of interferon signaling molecules, several chemokines, and other cytokines important to the development of the immune systems. And what this group found, together with other seminal works, showing that these autoantibodies may be involved with the severity of COVID-19, for example, impairing the immune response. But our question after this work, and during a moment when we came from Germany and to do our Jovem Pesquisador project here in Brazil, was that we characterized networks of autoimmunity associated autoantibodies. And then we were characterized autoantibodies in healthy subjects, and finding that this, for example, these GPCRs targeting autoantibodies, they were essential for maintain physiological and pathological immune homeostasis. And we were characterizing that. So we passed this approach in association with several co-authors from Germany, Israel, United States, and several other countries to analyze the cohort of patients with COVID-19 to see how autoantibodies targeting GPCRs, how they might be involved with the development of severe COVID-19. And then, as you can see here, and in agreement with our previous work, that patients with COVID-19, severe COVID-19, they have a loss of autoantibody normal correlation signatures. You can see here in controls, mild and moderate disease, that the pattern of correlation, the association between these autoantibodies are conserved, but during severe COVID-19, we see a loss of this homeostatic association. And then you see here how we see traditionally through linear regression that each of these spots, what does it mean? Each of them is like a linear regression, each line according to the previous slides that we saw. And what we see that patients with COVID-19, some gain, some in relationship, and other autoantibodies, they lost their relationships. And here, a wonderful work that Gabriela has doing now during her PhD, showing that autoantibodies, she, in a collaboration with these other researchers from other countries, Gabriela saw that autoantibodies linked with several autoimmune diseases associated with COVID-19. The full paper is already submitted is now under analysis to the Lancet group, the Biomedicine journal. And what's the approach that Gabriela developed using data analysis and systems immunology? She investigated more than more than 200 patients approximately. So, and then she, with using ELISA detection of autoantibodies in serum of these patients, she developed several bioinformatic approaches trying to find common and latent factors. She did the traditional box plot analysis to see what was significantly different between patients and controls according to severity. She did functional enrichment analysis of the autoantibody targets, and she went for correlation analysis, random forest, and principal component analysis, as I showed to you in our previous slides. And then Gabriela made a nice characterization of severe COVID-19 patients, showing that these individuals, they have higher autoantibodies. As you can see, I will show each of these spot in the heat map is here, for example. Let's show here, that's more clear. You can see, for example, that autoantibodies associated with antiphospholipid syndrome, like cardiolipin, autoantibodies, they increase their levels according to disease severity and accompanies the immune response. What is very interesting, that the anti-SARS-CoV-2 antibodies that are protective against severe diseases that also increase, which Gabriela saw that all other autoantibodies found in patients with COVID-19 targeting neuronal molecules and also involved in cognition, synaptic plasticity, memory, which is really important to understand. For example, the long COVID symptoms that we have been seen associated with COVID-19, and she characterized well that these molecules are associated with COVID-19 severity. And she saw in the paper, just to show another follow-up, that our PhD student, Denis Leandro, who is doing wonderful relationship analysis and finding that some of these autoantibodies characterized by Gabriela, they have a strong age association compared with controls. You can see here through this linear regression, each of these green spot here in this heat map, just to illustrate. Denison found together with Gabriela that in COVID-19 patients, we have a strong association, a statistical significant association with age. The same Igor, who is doing his master student PhD, his master studies here in our lab. Also, he found also several other autoantibodies, anti-GPCRs, that are also associated with age. So, taking together these autoantibody histories that Gabriela and Denis and Igor are studying, we have seen that there is a strong breakdown of self-tolerance, which is explained by several papers that are coming up, because the SARS-CoV-2 has a strong molecular mimicry with our body, and also several other mechanisms like bystander activation, the breakdown of homeostatic inflammatory process. So there are several processes that will render for us years of investigations to understand specifically why we are seeing these results in the context of COVID-19. In addition, just to illustrate, here is illustrate some COVID-19 studies during the pandemic. Carol did multi-omics investigation, wonderful work developed by Carol. She compared data sets of proteome and also transcriptome analysis, and she found groups of genes in common between several data sets, global data sets in blood, and she found some in common in swabs. And Carol searched for exoma data. She got some Brazilian patients, and she did some analysis. And because she were finding several cell cycle associated molecules that are dysregulated in COVID-19, which can explain, for instance, the cellular alteration that we showed in the beginning of our presentation. And Carol found structural alterations in several genes, cell cycle associated genes. I will just show an overview of several genes, common genes between some Brazilian patients, and she's now preparing the manuscript that we hope to show you soon, this data in more details. But this, what we have sit down in our computers, in our balconies, or when the labs were unfortunately closed during the COVID-19 pandemics. We were mainly focused on COVID-19 research, but in parallel, there were several other works going on in our lab using integrative systems immunology approach to understand other and old diseases, for example, representing important chances, like Desiree, who is a PhD student from the Department of Pharmacology. She is doing a great job with data sets from dengue. She gathered data sets from all data available for dengue regarding to transcriptome analysis, and she tried to do some integrative analysis to understand going from differential expression analysis, integrating this data, trying to validate and compare molecules which are important to predict the development of vaccination, trying to characterize how the vaccination trials for dengue, they overlap with the natural immune response. So Desiree already found, using a systems immunology approach, several genes, interferon-associated genes, and genes important for the defense against virus, including the type 1 interferon signaling associated genes, as you can see here in the circle plots, several up and down regulated genes in common between these vaccination trials, which we hope will bring up more chance to cure the disease, and what the people are trying around the world after the challenge that have been faced by different groups to develop the dengue vaccination due to the autoantibody enrancement, this enrancement of the disease, and then particularly using the two data sets of the two trials, vaccination trials. This year found 28 genes, actually 26 from one, 11 from the other, but when merging the analysis, we found 28 genes here, mainly associated with type 1 interferon, which are modulated during early disease, early acute disease, and during late acute disease, the next days after 3-4 days of infections, and also in the convalescent phase. Desiree made the characterization. The paper is almost ready for submission, and we hope to show in more details these last works that we are showing to you soon published. No? But also, in addition to dengue, COVID-19, and several other diseases in context, Ranieri, for example, used integrative systems immunology approach during COVID-19 pandemics to characterize the immune response against Candida species. So here an example, he analyzed Candida glabrata also and other subspecies, but here just to show the overlap between Candida albicans and auris, which was characterized to be resistant to treatment, no? And which really important to find these targets. And Ranieri found that there is a wonderful systemic relationship between these toll-like receptor genes and interferon-associated genes. The paper was recently published. Everyone who wants to see more about this, but he saw groups of toll-like receptors and interferon-associated genes, just to show you the importance of finding this, not just to do descriptive analysis, but try to do some mechanistic understanding, not in a linear and Cartesian way, but to understand this, seeing several molecules simultaneously. What we did, Ranieri saw that the immune response to these genes, in one side we have the interferon-associated genes here in the y-axis, and the x and the other here axis. The x-axis, we see the toll-like associated genes that we see a not strong relationship, a moderate relationship between these genes, but under activation, upon activation, using Candida albicans, what we see that these genes, they cluster to activate the immune response. This is an example, a strong example, that what we see about the immune response, that not just single molecules are mechanistically important to understand how the immune systems work, but the immune system in this paper wonderfully published in Nature Immunology shows and that the immune systems are bringing up the theory of the network theory of the immune system. That, for example, some molecules, they're just actors or some immune cells, and they have their role specifically in context, in some tissues. And they have several intra-layer communication, also inter-layer communications that works dynamically to understand how the immune systems function in real life. And here, just to conclude to you, like what we have done during this pandemic, we have faced a puzzle of several information going very quickly throughout the world being generated, and we made our best doing to this hard exercise to find some important information through this group here that we formed in the Department of Immunology, that we have our postdocs and associated research, to whom I'm really, really thankful. I had the pleasure to get to know these people during pandemics, with this great opportunity to work with them, particularly Dr. Paula, who has done a wonderful partnership with me, working together with the students, Lena, Alexandre, who is a statistician, who have given a lot of support to us. And here, our PhD students, Denison, Gabriela, Desiree, who had worked a lot during this pandemic. Also our master students, Igor, Carol, Ivana, Ranieri, and the scientific initiation people like Julia, Lucia, Duane, Aline, thank you so much for being with us here, just to show our designer, Marcos, who helped us also with the presentations. And of course, our extremely thanks to FAPESP to support, to give us the Jovem Pesquisador project, and also the COVID-19 project that we did our best to develop a good work. And here, my thanks to the Department of Immunology, to Pharmacology, and to all our research collaborators throughout the world, Professor Renzox, Condino, with whom I did all my graduation studies, and many researchers who have shared data with us, ELISA data, which we use to develop our works, and also several others that we get to know before pandemics and also during to the pandemics. And here, to finalize, soon we have our biannual subject that we call Systems Immunology, and then we call this next semester is Integrative Systems Immunology. My thanks to all who have participated with us of this history during the pandemics, and I hope that's all. I hope we could pass the message what we have done during to these pandemics in our house working with Integrative Systems Immunology. Thank you, Paula. Thank you, Bruno, who I think was Bruno who did the kind presentation, introduction of myself, and Paula also. Thank you very much, and it was a pleasure. Thank you all for being here and to participate, all the people who are seeing our talk.
Thank you so much, Dr. Otávio. for your amazing talk. There's a lot of data, there's a lot of papers, and now I want to open for questions. So please feel free to open your microphones or send the questions in the chat or via YouTube. If you are via YouTube, you can send the question via YouTube. Thank you so much. I don't know if Bruno wants to say something. Bruno, we are doing our webinars together within Federal University of Rio de Janeiro. So we are also have the students from here.
Fantastic. So
thank you for all
Great.
I just want to thank very much Dr. Otávio Marques for a wonderful presentation. I think everybody is a bit in shock with all the data, but there is a question that is coming up on chat. There will be a question soon, but Paula, if you have any questions over there, maybe you could start.
I have a question. So maybe we saw a lot of data. So my question is about how can we know that all this data came from humans, and what do you think about what is the ways that you have to prove all this data or to validate all this data? If you have some idea, what is the best way to validate all this data in a small cohort, or how can we apply all these molecules, all these genes in the context of the disease?
A great question, Paul. Thank you very much. For instance, just to tell you first one example of our group, the paper from Tabata that we show some neutrophils-associated signatures in patients with deletion of CD40 ligand. What we did, we investigated patient missing one of these molecules, that was the CD40 ligand, which I was investigating during my PhD and master in the Department of Immunology. And then we saw a cascade of changes. What we see that we change one point and we have a domino effect, seeing that several molecules. When we are seeing a single change in our system, actually, we are missing many other events that are occurring. So our body is really dynamic and systemic, and then it's necessary to look this mechanistically and linearly. That's really important, but we have to look them also holistically. This was one example, but another one, just to conclude quickly, was the group Aikiko group in the United States, in Yale. She did she transferred to mice one particularly autoantibodies that were predicted to be pathogenic, and then she saw really the exacerbation of the disease. The disease severity was increasing in the mice. We did also now one of our papers from 2018 in Nature Communications. We saw one autoantibody which was central to the interactions, the homeostatic interaction between the autoantibodies, and then we immunized the mice through a collaboration with a group from Cell Trend, and then we overexpressed one of the autoantibodies, and then the mice was developing severe disease, like was another approach, was with systemic sclerosis, but were similar pathological events occurring in this disease, what we are seeing now with COVID-19. So, summarize our response, there are several ways to do, ranging from cellular models to mice with CD40 ligand mutations, with point mutations, and to also patients, clinical trials which have used blockers of particular molecules, as they are doing with Elan and other neutrophils associated molecules like CXR2, for example, which have been blocked by antibodies or by small molecules. So there are many ways to do this. And great question, Paul. Thank you very much.
Thank you so much, Dr. Otávio. Professor Julio, please, you can.
Otávio, it was, Paula, really congratulations. I think it's é uma mudança realmente de paradigma, né? We have been trained the whole life in systems in which you call Cartesian. So it is difficult for us at this stage of life, to start again. But it's tempting to interact with you. My first question may be related to this very interesting example that you just mentioned right now, because it is part of your own journey, CD40L, And I remember that the last paper that Ralph Steinman published before he actually died, I think it was a PNAS paper at that point, calling our attention to the interaction with platelets. And platelets and dendritic cells. So platelets as activators of dendritic cells. Since you mentioned this case, which is very interesting, of a patient that have that deficiency, I just wondered, what the clinical information that we know in clinical immunology about these patients, so that we could at least, give some thoughts. Paula, she's right, because she's saying, basically, we have, a universe of information here. They're very exciting, but how can you move from that to study cause-effector relationship, right? So you have to go to animal models.
Yes.
And dengue is very complicated because you don't even have an animal model that could be useful to test and validate some of the concepts that were mentioned there. So you're very limited to the people that have genetic deficiencies in order to, to make a rationale. So let me hear what you have to say. And before finishing, because I don't want to take too much time, so many colleagues would like to make questions. At some point, maybe Paula and you could discuss with our group, because we have now several years of experience in the hamster model, in which we study the integration between immunity and the microcirculation. And we never had an opportunity to do integrated immunology or pathophysiology in the system. And I think it could be a very, very interesting opportunity for you. And it would be so nice if we have a course and then the course leads to an interaction that we could never imagine before. So this is basically the message that I would like to leave now.
Oh, thank you, Julio. Thank you very much. It will be my pleasure to interact with you and the guys there. Thank you for the opportunity to clarify even more what Paula asked. I will give a practical example with CD40 ligand. We first did RNA sequence analysis of neutrophils from patients with CD40 ligand deficiency. What we are seeing on that time, patients with CD40 ligand deficiency, you may know, they are characterized by a hyper IgM syndrome. And classically in immunology, was known as a B cell defect. But then the patients, they receive, let's talk translationally for the clinics, the patients, they were receiving immunoglobulin replacement therapy. And they were, even though, dying doing to that. And then we had the question, which pathological process is still going on in these patients? And then what we did, we characterized the myeloid cell defects, and particularly we went to the neutrophils. And then we got the neutrophils, we did the RNA sequencing analysis, and then we saw several neutrophils-associated genes involved with neutrophils development, with hematopoiesis, with myeloid differentiation. And what we did the next, I got this was during my postdoc also, with transition from postdoc in Brazil to GM. And then we got AGL60 cells, which are cells, promyelocytic cells. We activated them with soluble CD40 ligand. And then we saw first in the cell line that the CD40 ligand has an important role during the development, differentiation of the promyelocytic cells. And then Tabata got this work. She did her PhD focusing on CD40 ligand deficiency. She continues investigating neutrophils defect to better characterize. She went to United Emirates University during her PhD in a collaboration with another group from associated with us, and she got bone marrow from mice with CD40 ligand deficiency. And she saw an array of alterations during development, comparing bone marrow neutrophils with wild-type mice. And then we went from cell lines, mice, and then we returned to the clinics, and then we saw that several patients not responded to only to the immunoglobulin treatment, they required GCSF replacement therapy called granulocide. So then these patients, they show a much better prognosis. And then we went systemically to understand that they're classically well known from 1993 when the disease was discovered, and in 1992 when the CD40 ligand was discovered, initially expressed by T cells, that this molecule, the soluble CD40 ligand expressed by platelets, as you pointed very well, goes to the periphery, circulated throughout the body, explaining some neurological dysfunctions and explaining why in the bone marrow environment, in the milieu of the environment, these molecules control the production of several growth factors, and also which in turn act on the cells, indirectly showing the function of CD40 ligand, but also directly CD40 ligand control systemically. That there is a paper in 1997, excited, that I saw that's called CD40 ligand in the center of the immune universe. And this was, it's very nice. You can put in your file. I think it's 1997. And then I was getting more into the systemic world to understand, for example, from insights to cells, in vitro, to mice, going back to the clinic, and understanding a context in a holistic way. I hope if
no, it's a wonderful story. We could keep discussing this for another hour. I think that there are many people probably in the audience that want to keep on, so I don't want to take more of your time. I was very curious about the autoantibodies. I had a problem in the communication. I lost 10 minutes of your talk, and it was terrible because I don't know whether they were IgM, IgG. I don't know. I don't know if the antibodies that you have so many antibodies against GPCRs, if they are known to work as agonists or if they work as antagonists. I saw bradykinin receptor in your list. I saw ACE2 receptor in your list. So, I mean, we could discuss this for another hour. So let's move on, but
okay. They are agonistic, Julio. Several of them, they are agonistic. Like they are now testing them in Germany. they see that they induce production of important pro-inflammatory cytokines, and they are associated with the endothelial damage. So there are several working now mechanistically going on from our insights, proving that most of them, they are agonistic, but this is another point that we have to see, but because maybe some are antagonists, like the interferon type 1, the autoantibody target interferon type 1. But we can make a course together. We can. We are very open to discuss more this excited field. Thank you so much for your interest. I'm very happy.
Oh, but we have to thank you. I mean, it's
thank you so much.
Broadening our vision about systemic immunology. Thanks so much.
Thank you, Julio. Thank you so much for your interest.
Thank you so much, Professor Julio. Now, Bruno, do you want to make a question or make the Bianca?
Yeah, that's what I was going to do. We have a question from one of our PhD students, Bianca Prade, that asks how the age-related genes were selected and what was the median age of the patients.
You mean of which study? You mean from the autoantibody study or from the Paulo study for the DAGs, the dysregulated genes? Bianca? So anyhow, I can tell from both if you want. We had patients ranging from 30 years age, right, Paula? Paula can correct me. Until old, as not old patient, but 70 patient with some age older, I would say, with 80 something years of age, and then we search for autoantibodies which were and genes which are differently from young to old patients, and we get each of them like in a binary analysis group below 60, above 60 based on rheumatological parameters, and to characterize better these genes, these autoantibodies in a more continuous spectrum, like to see then through regression analysis that Denison is doing very well now. And then Paul and Denison went deeply on these aspects with Alexandre also to then characterize. We first do a screening to see which are statistically different, and then we go deeply to each of one to see precisely how they behave according to the age in a continuous line. The way how we have done. If there is any new or other approach, we would be very happy to know, and it will be very welcome. So.
Yes, great. And all the data regarding the median of age, we publish it in the supplementary data also. You can check all the age of the groups in the supplemental data.
It's a good point, Paula. Good that Paula brought it. There's sometimes so difficult to get this data. And what we do is after getting all of them, which is hard work, the people have nicely worked there in the lab. I'm very thankful to the members of our lab that they make all this data available. If you go to the supplementary data of all these papers, they have several supplementary tables supporting each figures. Also, all the codes available, we make them available to the people to reproduce the figures, also together with what the supporting TXT table to anyone run this with this code. And if anyone has difficulties or needs to help for this, please contact Paula, me, or any of our lab members that will be more than happy to support this field. No?
Yes, thank you. And Miriam, Professor Miriam, have a question?
I'll try to ask.
Miriam, tá mutada.
Sorry, okay. We had some microphonia here, so okay. So now I think it's okay. So first of all, thank you, Otávio, for the data you presented. It's really, really nice work. And I'm very curious about this sex bias on the infection of COVID-19. And also from what I've seen, like I have some friends that was exactly that, but the male part gets sick and the woman even like sleeping on the same bed and all doesn't. But at the same time, once she gets sick, usually like the woman had a worse symptoms than the man. And I was wondering, I haven't seen any data on separating death from like women and men, only infection. And I wonder if you did see in your cohorts or in the data that you analyzed, if you see any differences, like after getting sick, if women have worse development of the disease, and if you do have a higher death toll on women than men. Also thinking about immune diseases that usually, like some of them are women get more than men. So I was wondering a little bit on that end, if you have that data.
Great question. Paula did this nice job. And we see that women, they were developed less severe, but we don't have data. Paula moved forward now to work with cardiomyocytes, but we didn't have follow-up data on women who develop severe disease compared with those which not. That's a very good point, Miriam. Thanks so much for your question. That we can think about, Paula, for some students to investigate it, to understand the reasons for these differences. But I really do not have data from women with less severe. Was this your question, if I understood well, Miriam?
No, it was about the severity of the disease and like and number of deaths. If you'd have like a more severe disease and more dying women than men because of that. Because if you look at some autoimmune diseases like lupus or some others, women have more because apparently like could get a more persistent or a higher inflammatory situation. And since in some cases that will be the cause of death in COVID-19 would be like the response after infection. If you could have a protection to be infected, but once you are infected, you get worse disease for women. Yeah.
Let's see if I understood well, because there's a little bit echo, but if I understood well, yeah, when the woman they get infected, they tend to develop less severe disease with the men in general, but it's a tendency. But there is many other factors that can lead to a severe disease. We have seen this, but one of the aspects that we saw some protective genes that women are able to upregulate it better when they get infected than the men. And also they are able to downregulate a set of genes associated with neutrophils that further we characterize that these genes are associated with tissue damage. And what's happening? Women, they get infected as men in the same proportion, but after when they get sick, or they develop a more severe disease, in some cases, they have an intrinsic capacity to modulate the immune response to combat the virus, but in simultaneous try to reduce the damage. I will make just an analogy to make easier the understanding, like a car which is able to speed up very quickly, but can brake better. So a woman, they are like cars which are able to speed up quicker, like getting a strong immune response, but they are able to break the immune response in a better way, making protections. And one of the other systemic analysis we started to do, but we have started to get some data sets, but we didn't finalize this analysis, was just comparing them with systemic sclerosis, lupus, arthritis, and several other autoimmune diseases. We have a lot of interest to do this, not only for COVID, but we are thinking to integrate data from viral infections into the intersection between viral infections and autoimmune diseases that we hypothesize that's much more straight and close than we have in the literature. There are possibly several other viral infections that are involved, considering the interaction between virus and our immune response. So, in our body, the virus, they have the property to integrate to our cells and being there genomically and passing from generation to the other. And in addition to our microbiome, that we are more prokaryotic than eukaryotic, we have also several transposons and several viral materials inserted in our cells that probably are influencing these outcomes of COVID-19 and several other diseases. Miriam, I don't know if it was this point. If not, Paula, please can help me. Miriam, if
I would like just to complement Miriam, because in the last year, we used the data that we get the data from a database called the Global Health 50/50. And it is a database that you can have the information about incidence and death rate about men's and women's about the COVID. So in this database, you can see that the death rate is in the majority of the countries is higher in the men's. So the death rate is higher in the men's. And there's a lot of theory about why we have high incidence of mortality in men's. And we just bring a little piece of information about the neutrophils, but the information about the mortality, the high mortality in men's, you can get inside this website, this database called Global Health 50/50.
Okay, thank you.
And also we have Bruno, Professor Bruno, we have more questions.
Yeah, I just want to first, I think, give my 2 cents on Dr. Julio's question about the lower African incidence. And it was shown recently in a study in the University Hospital morgue in Zambia that Africa has no lower incidence. What it actually has is massive subnotification of cases and deaths. No, yeah, and but it's another issue. But what I was actually going to ask is how to deal with the massive anxiety attack that I had when I saw that much data, and I was actually zooming at my phone screen to actually try to look through each gene code and see if I can actually identify any of those genes. And every once in a while, I would see one that I know this one. And how someone that was trained in the more linear approach can actually make this jump to systems biology and systems immunology specifically.
Bruno, you had a paradigm crisis.
Exactly.
I think it's an existential crisis.
Existential crisis. this is also true for our psychological life, Bruno, Julio. Life is this: we are immersed in a myriad of information, and we look only pieces. There are some parabolas that call each person gets a mirror, a piece, and think that they have all the knowledge, and just we have pieces of information. And in the end, we only know that we know nothing, Bruno? But that's the point about the systemic information. We have to look to try to understand relationships, how these relationships behave. Of course, the mechanistic studies, they are very important to understand in details how these molecules are there. But the message, the take-home message is, look, we are just seeing a piece of a small part of everything that's going on in our life. When we are judging a person, when we are judging a cell, when we are judging a tissue, a whole organism, there is a holistic world going on behind. I think this is the second part of your crisis. Bruno, I started to do psychological therapy. Last 4 years, I do psychological therapy to get to know myself a little bit better, try to get in order with my emotional chaos that appears sometimes with my life. That's one of the points I always suggest. Do this for each of one for personal development to reduce the anxiousness. But about the other point is about the Africa. Why Africa is getting less case, we don't know, but one of my hypotheses is the hygiene therapy, hypothesis, no? That this has a high impact on immunomodulation, and that might be one possibility to explain this. But of course, we need several studies to define this point. For example, I agree that masks are important for protections. I really agree that hygiene is important, but the studies show that we have to think also. My mother always told, water in excess kill even plants. So even these non-pharmacological approaches to protect against COVID-19 must be taken carefully. For example, how long we can survive with less oxygen without damage in our brains or future damage in old age, when we have a lack of full breath in and an excess of breath out of oxygen and carbonic gas. So we have to take care of all of the things, how we deal to take care, to be moderated, to scientifically in agreement with science, in agreement with science, to protect ourselves, to avoid the spread. But until when we need to continue with these measures, and for how long time this will not damage ourselves, like being with excess of cleanness. Kids, there are several studies showing we have to go to nature, we have to walk barefoot to get contact with earth, we have to get contact with plants, we have to get contact with humans. So we are a social species. The human being is a social species. We have something that's called exposome in systems immunology. That is all environment material that we are exposed. And this significantly modulate our body, ourselves, even psychologically. So this is another matter that we have to think when explain why in Africa, for example, is less susceptible. It's just devaneio, né? Just a elucubration of and about why maybe this is a possibility. It's something that we have to do also. We have to think here about ourselves. We are living, they call in stones forests, né? So we have to be in contact with nature. We have to make ourselves stronger, be in contact with human beings, just trying to remember to going back to be human being after pandemics, not forgetting that we are human beings.
Great.
I don't
Thank
know
you
if
so much.
this that Bruno want to ask.
I don't know.
No,
What's
just
this?
to say thank you very much. It was a very nice talk, very thought-provoking, some anxiety-provoking, but we are going to deal with it. And but thank you very much. I just want to tell everybody that next week we are going to have a talk by Dr. Dan Dwyer from Harvard University that will be talking on the effects of TGF-beta in the differentiation and activation of intraepithelial mast cells. So for those interested in mast cells, it will be a talk from someone that is one of the most interesting and youngest people in the field. But for those interested in immunology in general, it will also be a very interesting talk on the microenvironment that regulates the immunological reaction. So, Paulo, please.
Bruno, you know, when I saw this written that TGF-beta regulation of intraepithelial mast cell, I was in crisis here also, Bruno, because I thought, what is this question for me? How I can, how you explain this? I don't have data with mast cell. And then I saw, oh, now it's clear.
Yes,
So,
it's our next week webinar.
Next, a very mechanistic, linear approach to going back to the Newtonian, Cartesian way, Bruno, of life.
Obrigado.
Thank you so much. Thank you, all the students and all the professors that participated in this immuno webinar. We did a great immuno webinar. Thank you so much. And we see all of you next week. Thank you so much. Thank you so much, Professor Otávio, for this great talk.
Thank you.
Yes, thank you
very much. Everyone who participated, it makes me really happy to be here with you guys.
So, see
Boa
you
semana para todos.
Thank you, Professor Julia. Thank you, Professor
querida.
Bruno, Miriam. Bye-bye.
Tchau, bye.